Greetings, dear learners. Welcome to the official YouTube channel of SRN International School, Jagatpura, Jaipur, Rajasthan. I am Faculty of Chemistry at SRN International School. Today, in this video, I will be explaining Unit 14, Biomolecules of Grade 12, Chemistry, NCERT. A living system grows, sustains, and reproduces itself. The most amazing thing about a living system is that it is composed of non-living atoms and molecules. Living systems are made up of various complex substances called as biomolecules like carbohydrates, proteins, nucleic acids, lipids, etc. So let us start with carbohydrates. Carbohydrates are primarily produced by plants and form a very large group of naturally occurring organic compounds. A carbohydrate may be defined as optically active polyhydroxyaldehydes and ketones, or the compounds which produces such units on hydrolysis. One of the examples is glucose. Carbohydrates include sugar, starches, and cellulose. Function is to store energy and to provide shape to organisms. They are composed of elements carbon, hydrogen, and oxygen. Glucose is the example of carbohydrate. Its formula is C6H12O6. Molar mass is 180.156 grams per mole. Melting point is 146 degrees Celsius and its heat capacity is 218.6 kilo per Kelvin per mole. Joules per Kelvin per mole. The open structure of glucose is a straight chain with the first carbon having an aldehyde group, that is CHO group. But the beta D-glucose is a cyclic structure of glucose which has a pyranose structure. So it is called as glucopyranose. Carbohydrates are classified on the basis of their behavior on hydrolysis. They are monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are those carbohydrates which cannot be hydrolyzed further into simpler units of polyhydroxyaldehydes or ketones. They are, for example, glucose, fructose, and galactose. Disaccharides are the carbohydrates which can be hydrolyzed to give 2 units of monosaccharide. For example, sucrose, lactose, and maltose. Oligosaccharides are the carbohydrates which can be hydrolyzed to give monosaccharide units from 2 to 10 units, like for example raffinose and stachyose. Polysaccharides are the carbohydrates which on hydrolysis gives more than 10 monosaccharide units. They are like for example starch, glycogen, cellulose. Carbohydrates are also classified as reducing and non-reducing sugars. Reducing sugars are those carbohydrates which act as a reducing agent, and it is due to the presence of aldehydic or ketonic group. Whereas non-reducing sugars are those carbohydrates which cannot be act as a reducing agent, and because they don't have any free aldehydic or ketonic group. The reducing sugars are the sugars which will give a positive test with the Fehling solution, whereas non-reducing sugar will give a negative test. And in case of reducing sugars, the examples are like monosaccharides units, all the monosaccharide units, and some of the disaccharides. Whereas in the non-reducing sugars, the examples are like some of the disaccharides as well as all the polysaccharides. So example is like sucrose. Sucrose is a non-reducing sugar. Preparation of glucose. Glucose are prepared from sucrose, that is cane sugar, with the formula C12H22O11. So sucrose is boiled with dilute HCl or H2SO4 in alcoholic solution, and we get a glucose and fructose in equal amounts. It can be also prepared from starch by hydrolysis of the starch in presence of acid. Starch is C6H12O5 enzymes to give n moles of C6H12O6. That is, we get the glucose from starch on hydrolysis. The reactions of glucose. Glucose reacts with various reagents and its structure can be elucidated from these reactions. So glucose reacts with hydrogen iodide, that is HI, to give N-hexane, normal hexane, which is a straight chain carbon, indicating that the glucose has all the 6 carbon in a straight chain. Second reaction is with hydroxylamine, NH2OH, and it gives glucose oxime, where the first carbon is converted into oxime. This shows that glucose has a carbonyl group. Carbonyl group is C double bond O. Similarly, another reaction to show that glucose have a carbonyl group is with HCl, hydrogen cyanide, where it gives cyanohydrin. The third reaction is that when glucose is reacted with bromine water, so it gets oxidized and it forms gluconic acid. This gluconic acid has a COOH group at carbon 1, showing that glucose has aldehyde group in carbon 1. Similarly, glucose also reacts with dilute HNO3, a strong oxidizing agent, which forms saccharic acid. The saccharic acid has a 2-carboxylic group, COOH group, at first and 6 carbon. Then the last reaction is with acetic anhydride. When it reacts with acetic anhydride, then it forms glucose pentaacetate, showing that glucose has 5 OH groups. The second biomolecule is protein. Proteins are most abundant biomolecules of the living system. Chief sources of protein are milk, cheese, pulses, peanuts, fish, meat, etc. All proteins are polymers of alpha amino acids. Alpha amino acids has a carbon which is alpha due to the 2 functional groups attached to it. One is COOH group, which is carboxylic acid group, and the other one is NH2 group, which is the amino group. So therefore this carbon is an alpha carbon. So it has alpha-alpha structure. Now when the 2 amino acids, they combine together to remove water, then there is a bond formed called as peptide bond. And this peptide bond is CONH bond, and thus these amino acids, they combine and remove the water to form a polymer called as protein. Structure of amino acid. Amino acid has a carbon to which a carboxylic group is attached and an amino group is attached. This carbon is alpha carbon, and to this again a hydrogen is attached and then an R group. This R group is a variant. That means the R can be hydrogen or it can be CH3 or any other group. When it is hydrogen, then its amino acid is called as glycine. If it is CH3 group, then it is called as alanine and so on. So there are 20 amino acids known. Now here, the carboxyl group is shown as COO minus, which means that H plus of the COH group is now attached to NH2 group, that is amino group, in the form of NH3 plus. So there are two plus and minus charges on it, and such type of dipolar molecule is called as Zwitter ion. So amino acid is an example of a Zwitter ion. Proteins are denatured. So these proteins are found in biological system with a unique 3-dimensional structure and biological activity, and it is thus called as a native protein. But when it is subjected to some kind of physical change, for example temperature change or a chemical change, like for example pH change, then the hydrogen bond, it gets disturbed, and thus the globules, they unfold themselves or into a form of a helical structure which is uncoiled. So this process of protein is called as denaturation. So in denaturation, the um, the protein gets uncoiled, but its primary structure remains intact. It can also get renaturation. That means again it can fold together to give a tertiary structure, and that is called as renaturation. The third biomolecule is nucleic acid. Every generation of each and every species resembles its ancestors in many ways. It has been observed that nucleus of a living cell is responsible for this transmission of inherent character called as heredity. The particles in nucleus which are responsible for heredity are called as chromosomes, and these are made up of protein and another type of biomolecule called as nucleic acid. Nucleic acid are of two types: deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. DNA is usually it occurs inside the nucleus and some cell organelles, whereas RNA is present in cytoplasm. DNA is a genetic material, whereas RNA is not a genetic material, and it is except in case of certain viruses. And DNA is a double helix structure. It is double stranded actually. It is a double stranded. Two strands are there, whereas RNA is single stranded, with some exception of some viruses. And DNA has got sugar called as deoxyribose sugar, whereas RNA has a ribose sugar. Nitrogenous base in DNA are adenine, thymine, cytosine, and guanine, whereas RNA has um, uracil in place of thymine, and other three are similar, that is adenine, cytosine, and guanine. DNA actually has, in the DNA, we find that there is a hydrogen bond formed between the complementary nitrogen bases on the opposite strands of DNA. That means adenine will have a hydrogen bond with thymine, and cytosine will have it guanine. Whereas base pairing is seen in RNA is seen in the case of only of the some of the coil parts, only in the coil parts. Nucleic acids are long chain polymers of nucleotide, as they and therefore they are also called as polynucleotide. Complete hydrolysis of nucleic acid, that is whether it's a DNA or RNA, it yields a phosphate group, that is phosphoric acid, and then it gives pentose sugar, and also a nitrogen containing heterocyclic compound, which are also called as nitrogenous base. Now these 5 carbon sugar, that is pentose sugar, are of two types, that is ribose and deoxyribose. So ribose sugars are present in RNA and in deoxyribose in DNA. This second carbon of the RNA is having OH group, therefore it is ribose, but in case of DNA, there is no OH, the oxygen is absent, and therefore it is deoxyribose. The nitrogenous bases are of two types, pyrimidines and purines. Pyrimidines are cytosine, uracil, and thymine. Uracil in RNA and thymine in DNA. Purines are of two types, guanine and adenine, and these purines are larger than pyrimidines. In the structure of nucleotide, there is a unit formed by the attachment of a base to one dash position of pentose sugar, and this is known as nucleoside. The sugar carbons are numbered as 1 dash, 2 dash, 3 dash, etc. in order to distinguish these from the bases. When nucleoside is linked to phosphoric acid at 5 dash position of sugar moiety, we get nucleotides. Dear students, I hope you must have understood this topic. Keep learning, keep exploring. Thank you for watching. Please like, share, and subscribe to our channel for more informative videos.